This video is going to use the last one as sort of a stepping stone into new topics. So as the last video talked about what a pacemaker cell action potential looked like, this video is going to talk about how action potentials produced at the pacemaker are distributed through the heart through what's called the conducting system. If you look at what's published in the literature, I found sort of contradicting opinions as to what the conducting system is, whether it's nervous tissue or modified muscle tissue. Personally, for the purposes of my students, it doesn't matter which one is right. What matters is what the conducting system does, and it is the network of cells that produce APs spontaneously, the pacemakers, and then distribute them through the heart so that we can get rhythmic contraction. To do this, I want to work with an image of the heart. So I have here a cartoon that I want to use. If this cartoon looks totally alien to you, you may want to go review cardiac anatomy. You should not be viewing this video if you don't know the anatomy of the heart already. But just as a benefit to everyone watching, I want to quickly orient you to what this little sketch is showing. In the upper left side of the picture, we have the right atrium. And we're looking at the heart from the front. So there's the right atrium, and below it, the right ventricle, with the tricuspid valve shown in gray separating them. On the opposite side of the screen is the left atrium on top and the left ventricle on bottom, separated by the bicuspid valve. In between the two ventricles is the interventricular septum. And I want you to notice on this diagram, in each ventricle, there are two papillary muscles, 4 total. 2 of them are labeled here, connecting to the chordae tendineae of the bi and tricuspid valves. Those papillary muscles will be important later. That's why a couple of them are being shown here for illustration purposes. So here's our heart, and let's just put the conducting system right on here. Conducting system, or the production of APs, begins at what most people would call the true pacemaker, which is the sinoatrial or SA node located in the upper portion of the right atrium. Action potentials occur there spontaneously faster than any other portion of the heart. That's why it serves as a pacemaker. Action potentials produced at the SA node spread out into the 2 atria. So it spreads out to the right atrium, it spreads to the left atrium, and directs those 2 chambers, the myocardium of those 2 chambers, to contract. Eventually, APs end up at the atrioventricular or AV node in the lower portion of the right atrium near the ventricle. This is the area in the heart that in essence has the second fastest production of action potentials spontaneously and will serve as sort of a backup pacemaker. That's all I want to say about that here. Going into more details is beyond the scope of what I want to do with this video. Action potential propagation at the AV node slows down quite a bit. The advantage of this is we want to have rhythmic contraction of atria and ventricles, and so by having AP propagation slow before the APs enter the ventricle, allows the atria time to complete contraction before the ventricles begin contraction. The AV node is connected to the ventricles through the atrioventricular or AV bundle, also called the bundle of His. It's pronounced His if you see it, but it's spelled H-I-S because it's a person's name. The AV bundle in a normal heart should be the only electrical connection between the atria and the ventricles. There are conditions where that's not the case, but you should also remember that the fibrous skeleton of the heart has as one of its functions to serve as electrical insulation between the atrium and the ventricles. So the AV bundle is where APs enter the ventricles, and you should already know this from having studied cardiac anatomy. The heart has a lot more ventricular myocardium than atrial myocardium. So while we had a slowdown in propagation at the AV node, we have an acceleration of AP propagation at the AV bundle because now we need to get the action potentials to both ventricles as fast as possible. Eventually, the AV bundles split into left and right bundle branches within the interventricular septum, the left branch for the left ventricle and the right branch for the right ventricle. And these continue to split to smaller and smaller branches until they end at individual synapses called Purkinje fibers, or individual extensions, called Purkinje fibers that synapse with the contractile myocardium. And notice the extensions that contain Purkinje fibers go up into the papillary muscles as well as the ventricular myocardium itself. The Purkinje fibers are far more numerous and fine in structure than I'm showing in this cartoon, but you get the idea that it's the Purkinje fibers that ultimately synapse with the cardiac muscle. And so transmission of APs through the heart begins at the SA node, spreads to the atria, spreads to the AV node, spreads to the AV bundle, to the bundle branches and the Purkinje fibers. And then it starts again at the SA node, and the whole process repeats. And that's how the conducting system moves APs through the heart. Don't forget that AP propagation speeds up at the AV bundle, it slows at the AV node, and tracing or tracking action potentials through the heart can be done clinically with the electrocardiogram, a diagram of which is shown here, and will be the subject of the next and final video in this series.